Hardy-Weinberg equilibrium. UGC-NET anthropology. Part 2. Welcome to AnthroNet Academy, a study aligned with the UGC-NET anthropology syllabus. Description box. Please visit the description box for books references. Dear learners, if you find this content helpful, please consider subscribing to AnthroNet Academy. Your support really means a lot and keeps me motivated to create more valuable content for you. Big surprise at 500 subs. Once AnthroNet Academy hits 500 subscribers, we're dropping something truly special just for you. A full video revealing 100 powerful, underrated, and must-read books list in anthropology, complete with author names and insights important for UGC-NET exam. Test yourself. Don't miss the final segment of this video. It includes high-yield MCQs that could make or break your next anthropology exam. 1. Inbreeding coefficient F. Definition. The inbreeding coefficient F measures the probability that two alleles at any locus in an individual are identical by descent, i.e., inherited from a common ancestor. Formula, simplified. Where N1, N2 equals number of generations from the individual to the common ancestor via father and mother. FA equals inbreeding coefficient of the common ancestor. N equals number of common ancestors. Key points. F equals 0, no inbreeding, random mating. F equals 1, complete inbreeding, all alleles identical by descent. F for offspring of first cousins equals 0.0625, 6.25%. Increased F leads to rise in homozygosity, reduction in heterozygosity, higher probability of recessive disorders. Anthropological relevance. Studying inbreeding helps understand disease prevalence, fertility patterns, and genetic health in isolated human populations. 2. Genetic load. Definition. Genetic load refers to the reduction in the average fitness of a population due to the presence of deleterious alleles compared to an ideal population with only optimal genotypes. Types. Mutational load, from new harmful mutations. Segregational load, from balanced polymorphisms, e.g., sickle cell trait. Substitutional load, cost of replacing one allele with a more advantageous one, Haldane's concept. Inbreeding load, due to increased homozygosity of harmful alleles via inbreeding. Formula basic. Where L equals genetic load equals mean fitness. Importance. High genetic load may threaten population survival. Management of genetic load is crucial in conservation biology and understanding human population history. 3. Genetic isolation. Definition. Genetic isolation occurs when a population does not exchange genes with other populations due to geographical, cultural, or reproductive barriers. Types. Geographic isolation, mountains, islands, deserts, etc. Cultural isolation, endogamy, caste systems, religious prohibitions. Reproductive isolation, differences in mating times, behavior, or game-tick compatibility. Effects. Limits gene flow. Leads to genetic drift, inbreeding, and possible speciation. In humans, can result in founder effects, genetic bottlenecks, and unique allele frequencies, e.g., Ashkenazi Jews, Amish Andaman Islanders. 4. Genetic distance. Definition. Genetic distance quantifies genetic divergence between populations. It reflects how different the gene frequencies are due to evolutionary processes. Common measures. Ney's genetic distance, D. D equals minus L Nye. Where I is the normalized identity of alleles between populations. FST, fixation index. Where interpretation of FST. 0. No differentiation. 0.05, low. 0.15, moderate. 0.25, high. Applications. Understanding human migration and evolution. Mapping ancestral relationships between populations. Forensic genetics and conservation. Genetic polymorphism. Definition. Genetic polymorphism refers to the coexistence of two or more alleles at a genetic locus in a population where the frequency of the least common allele is greater than or equal to 1%. This variation must persist over generations through heredity, not be a result of a recent mutation alone. Polymorphism literally means many forms and is a cornerstone of genetic diversity. Types of genetic polymorphisms. Type description example. Single nucleotide polymorphisms, SNPs, variation at a single base para G substitution in the beta-globin gene, HBS. Insertion/deletion, Indels, addition or removal of small DNA sequences for BP deletion in the CCR5 gene. Copy number variants, CNVs, duplications or deletions of larger DNA segments variation in the number of AMY1, amylase, gene copies. Microsatellites/STRs short tandem repeats of 2 to 6 BP, CA, and repeats used in DNA fingerprinting. Mini-satellites/VNTRs longer tandem repeats, 10 to 60 BP, used in paternity and identity testing. Structural polymorphisms inversions, translocations, chromosomal rearrangements inversion polymorphisms on chromosome 17. Mechanisms maintaining polymorphism. Balance selection. Heterozygote advantage. Heterozygotes have higher fitness than homozygotes. Example, sickle cell trait, HDS, confers resistance to malaria. Frequency-dependent selection. Rarer genotypes have higher fitness. Example, pathogens evolving to target common HLA types. Environmental heterogeneity. Different environments favor different alleles. Neutral theory of evolution. Proposed by Matu Kimura. Most polymorphisms are neutral, no effect on fitness, and persist due to genetic drift. Mutation drift equilibrium. New mutations introduce alleles. Genetic drift may fix or eliminate them unless selection maintains them. Functional versus neutral polymorphisms. Category characteristics example. Functional effect phenotype, often under selection ABO blood group alleles. Neutral no known effect on fitness, many SNPs in non-coding DNA. Significance in biology and anthropology. 1. Evolutionary insights. Genetic polymorphism reflects adaptive potential of a population. Maintains genetic diversity, which is key to long-term survival. 2. Population genetics. Polymorphisms help measure Genetic variation using heterozygosity. Gene flow, migration, and population structure. 3. Disease association. Many polymorphisms are linked to disease susceptibility, e.g., AP-epsilon-4 and Alzheimer's disease, e.g., HLA alleles and autoimmune diseases. 4. Forensic and identity studies. STRs and VNTRs are unique and used in DNA fingerprinting, paternity testing, forensic identification. 5. Anthropological applications. Tracing human migrations and population history, e.g., mitochondrial DNA, mtDNA, and Y chromosome polymorphisms. Understanding endogamy, isolation, and admixture. Reconstructing ancient human demography. Quantitative measure, heterozygosity. Observed heterozygosity, Ho proportion of heterozygotes in a population. Expected heterozygosity, he expected under Hardy-Weinberg equilibrium. Equals 1 minus 2. He equals 1 minus p. I 2. Where allele. High heterozygosity equals high polymorphism equals high genetic diversity. Examples of well-studied genetic polymorphisms. Polymorphism gene / locus relevance. HBS allele HBB malaria resistance, sickle cell anemia. ABO blood group ABO gene transfusion compatibility, disease risk. HLA polymorphism MHC region immune response variation. G6PD variants G6PD gene malaria resistance, hemolytic anemia. CYP450 polymorphism cytochrome P450 genes drug metabolism variability. Hardy-Weinberg and polymorphism. Under Hardy-Weinberg equilibrium, the allele and genotype frequencies remain constant in the absence of evolutionary forces. If polymorphisms deviate from HWE expectations, it may indicate inbreeding, selection, genetic drift, migration. Key takeaways. Genetic polymorphism is the basis of variation in all biological populations, maintained through natural selection, mutation, and drift. Provides tools for studying human evolution, adaptation, and health. Anthropology, genomics, and personalized medicine all rely on understanding polymorphism. Test your knowledge. 1. Which of the following would not qualify as a genetic polymorphism? A. A mutation with an allele frequency of 0.02%. B. A SNP with a frequency of 3%. C. An STR marker with 2 alleles, both greater than or equal to 1%. D. An indel with an allele frequency of 5%. Answer A. Explanation. By definition, genetic polymorphisms require a minimum frequency of 1% in the population. A mutation at 0.02% is too rare to be classified as polymorphism. 2. The persistence of the sickle cell allele, HBS, in African populations is primarily an example of A. Mutation load. B. Genetic drift. C. Frequency-dependent selection. D. balanced polymorphism? Answer: D. Explanation: heterozygotes, HBS, have a selective advantage against malaria, maintaining both alleles in the population A classic case of balanced polymorphism. 3. Which statement is false regarding polymorphisms and Hardy-Weinberg equilibrium? A. Polymorphic loci show variation in genotype frequencies. B. Departure from Hardy-Weinberg equilibrium always indicates selection. C. High heterozygosity suggests high polymorphism. D. Polymorphism can be detected in both coding and non-coding regions. Answer: B. Explanation: departure from HWE may result from many factors, not just selection, including inbreeding, genetic drift, migration, and non-random mating. Which form of polymorphism is most likely to be used in DNA fingerprinting? A. SNP. B. Indel. C. STR. D. CNV. Answer: C. Explanation: STRs, short tandem repeats, are highly variable and widely used in forensic DNA profiling due to their high discriminatory power. Thank you for watching. Please like, share, and subscribe to my YouTube channel, AnthroNet Academy, for more insightful content on anthropology. Stay safe and connected.